Yes, happy new year to everyone. So, welcome to the day 4 session on drug likeness properties, drug databases, and ADMET properties. So, we also discuss on yesterday's kind of tools and databases. Before proceeding to today's session, can we go ahead with the recap of last session topics as usual? What exactly did we discuss on yesterday? So kindly recap the last session topics. So Shritika Rangaprabhu, accept the invitation so that it will be easy for you to join. Okay, no one can uh, admit you in between. So take care. Okay, yeah. Just recap the last session topics. Yeah, I forgot. Is it? So I just showing you hint. So we discussed on. Yeah, I think you have taken the resolution for New Year. Very good. So we discussed on drug design methodology. We have discussed on different methods of drug design. You're right. And in before going to the types of drug design, I think we discussed initially traditional drug design and structure-based drug design. These are the two types. Then we have gone to the different terms, definitions, like what is drug design, what is drug discovery, what is drug development, and how about reverse stalking. And then in the methods of drug design, we have gone to Lipinski rule of 5, ADME, QSAR, especially QSPR. I told you QSAR is a subset of QSPR. So QSPR stands for quantitative structure property relationship, and if we determine activity in terms of property, then we call it as QSAR. And then we have discussed technical terms like heat, lead, so how to generate the lead and how to optimize the lead. And then we have discussed API, and then we have discussed IND, investigational new drugs, okay, NDA, new drug applications. And then we have discussed on phases of drug design. Okay, especially we discussed on molecular modeling, molecular docking, and molecular dynamic simulation. So in that modeling, we have recapped the three methods of target modeling, that is homology modeling, threading, and ab initio structure prediction. Then in molecular talking, we discussed on two terminologies, ligand and receptor, and we have discussed the theories behind. Okay, so this is what we have discussed on yesterday, isn't it? And especially we focused even on that in drug development, we focused on the four phases of drug design. Now, let me go ahead with today's session. Today's session, we'll be discussing on these topics. Before going to these topics, just I am providing an outline on about the university recognition. So I am PhD supervisor of Anna University Chennai. So I can able to guide the students of Anna University Chennai. That is those admitted in my institution. I can able to guide under because ours is an autonomous institution affiliated by Anna University Chennai, and I'm a research advisory committee member for many institutions, reputed institutions like Welltech, Rangarajan, Dr. Sekuntala R&D Institute of Science and Technology, VIT, Anna University Chennai, University of Madras, Sri Ramachandra Institute of Higher Education and Research, and also in Ethiraj College for Women. And even in this year, nearly for three scholars, I just appointed as research advisory committee member in Sri Ramachandra Institute of Higher Education and Research. Okay, so if anyone want to add me as a doctoral committee member or a PhD board examiner or any kind of eminent scientist member, and even if you like to invite for guest lectures, you are welcome. Now let me go ahead with the today's contents. Drug databases, we'll be discussing about these databases, drug likeness prediction, ADMET, and target prediction. First about PubChem. So in PubChem, I told you like we can either search through drug names, substances, or we can search through the compound names, PubChem ID. Also, we can give the keywords like genes, proteins, pathways. Okay, we have different kind of a keywords to search for any kind of a compounds in PubChem. Of course, that is the largest database for drugs and as well as natural compounds. Similarly, if you can download the 1D to 3D information of chemical compounds in ChemSpider. And Drug Bank is exclusively for drugs. So this is a fully annotation database for drugs. If you want to download any kind of a drug information, I mean, including the structures, we can able to get this information from Drug Bank. We have also so many tools present in Drug Bank. Drug Bank is not just a database. We also have many tools. Examples, if you want to check adverse drug reactions, like whatever drug we take and if it gives an adverse reaction or not, we can able to check from this tool present in Drug Bank. I'll be illustrating you in the subsequent slides. And CHEMBL. CHEBA is different, CHEMBL is different. So both are of course databases for chemical compounds. Binding DB we already seen. Even in cake, we have cake compound. In cake, there is a database called compound where you can download the information, 1D to 3D information of compound from cake. Like how you find the pathway, right, module. So like this, we can see cake compound. And CHEBI, I told you, this is also an exclusive database. Now let me go ahead with the drug likeness prediction. So in drug likeness prediction, we have different drug likeness filters. What is the purpose of going with drug likeness prediction through drug likeness filters? So generally, what route of delivery we prefer, I can give you the choices whether we want to take in the form of tablet or capsules in the form of oral drug delivery, or we want to take in the form of injections, intravenous, or we want to take in the form of syrup. In syrup, obviously, some kind of a sugar is added, isn't it? So the best form of a drug, I mean, we prefer to take only the oral route. Okay, just taking. So that's what you see. Many of the tablets, I mean, many of the drugs we take, we go with the oral route instead of taking in the form of injections. If so, if you want to check the oral bioavailability, okay, we can go ahead with this drug likeness prediction. Not only that, if you want to check whether any compound behaves like a drug or not. For example, if you want to treat cold, okay, especially cold and cough, which is the best natural remedy for treatment of cold and cough? So don't just rely on the allopathy medicines like Coldarin and other kind of strepsils, etc. The best medicine, if you ask me, in terms of a natural remedy, I can tell you the best is neem tree. The neem leaves are best enough to cure this kind of a cold and cough, isn't it? So if you see the neem leaves, we have more than 100 to 200 compounds. Not all the compounds can able to cure. Few compounds might be out of 100 compounds, 50 compounds can be extracted, and out of 50 compounds, 10 compounds show the potential, biological effect, and the remaining 10 compounds give a negative effect, antagonism effect, and maybe 30 compounds or neither giving this effect or that effect. Then our objective is to isolate just 10 compounds, and that 10 compounds, if we want to check whether it's going to have a oral bioavailability or not, and whether we can check whether it behaves like a drug or not, we can use the drug likeness filters. So here, the first drug likeness filters what we choose is the Lipinski rule of 5. You know why we call this as a Lipinski rule of 5? So before going to why we call this, I can also tell you this is also called as Pfizer's rule of 5. Pfizer, you heard about this? This is the second top important global pharmaceutical industry. The largest second largest global pharmaceutical industry is Pfizer, isn't it? So this Lipinski rule of five, also called as Pfizer's rule of five. And if you see this, okay, here in Lipinski rule of five, we have totally four properties. Number one is the molecular weight of the drug. It should be, so whatever we check initially, we check for a compound, and the molecular weight of the compound should be less than 500 gram per mole. And the second property is the partition coefficient, that is log P should be less than 5. The third is the hydrogen bond acceptor, which should be less than 10, and hydrogen bond donor should be less than 5. So these are the four properties of Lipinski rule of 5. Then why we call it as rule of 5? Because all these four properties are having a multiples of 5. Just because we have a multiples of 5, we call this as Lipinski rule of 5. And if you see this, okay, we also have few more properties like rotational bonds less than 10, polar surface area less than 140 angstrom. How we find these kind of properties, these are present in other drug likeness filter. Like how we find the Lipinski rule of filter, we also have Weber, Mugi. So there are types of drug likeness filters. So in each and every drug likeness filter, we have a different range, and even we have different physicochemical properties of the compound. Okay, so whatever the range values and whatever the properties they're fixed in this drug likeness, this can be used to assess the oral bioavailability as well as to verify whether this compound behaves like a drug or not. Drug, what do you mean by drug? Which gives agonism. Okay, agonism is a desired biological effect, and there is something called antagonism. Antagonism is not a desired effect. Okay. For example, earlier we are taking a drug called aspirin. Aspirin, also called as acetyl salicylic acid. This is a pain reliever. If you get a high pain, obviously we take this kind of a drug, aspirin. But this aspirin has yielded some side effects, especially with respect to cardiac problems. So this kind of drug, aspirin, which was taken for more than 20 to 30 years, this was banned, okay, completely, and now we can't able to see aspirin in the markets. So uh, the one what we check whether the compound behaves like a drug or not, we can use this. Am I audible to you, participants? Yes or no?
Yes, sir.
Ah, yeah. Thank you. Thank you. As I'm getting multiple messages, I don't. Okay, now let me go ahead. Um, if we go ahead, if we see this drug likeness filters, okay, we have these four properties, okay, and in the other drug likeness filters, we find the polar surface area and rotational bonds. Okay, now let me go ahead with a few more properties, but before that, I show you the softwares. MolSoft, Mol Inspiration, Swiss ADME. These are the tools we can use for predicting the drug likeness. And if you see the drug likeness, this can also be predicted from a database called IMPACT. This is an indigenous database. This is the database from India, a largest database from India. So IMPACT stands for Indian Medicinal Plants, Phytochemistry and Therapeutics. Okay, so if you feed a kind of a compound smiles format, or if you search for a particular compound, okay, in this case I have searched for a plant compound called vitaferin, which we also call it as steroidal lactone from ashwagandha. I got these kind of a properties. So instead of assessing through tools, we can also get the information from the databases. So one such database I'm showing you is the impact. This is an indigenous database of our nation. So like impact, even we have the well-known databases like NCBI, PubChem, and even most of the databases also have these physicochemical properties. So other than the physicochemical properties, we can see whether it's satisfying the drug likeness rule or not. Okay, even if you see in Lipinski rule of five, out of four properties, Okay, if we find any three properties are getting satisfied, still we can tell this compound behaves like a drug. Okay, so according to Lipinski, he has mentioned that even 3 out of 4 can we can term a compound as a drug. If you find 4 properties, then obviously no doubt it's a drug only. But if it is less than 3, that is 1 and 2 properties, then you can tell that compound won't behave like a drug. So in this case, we will be getting like failed. Okay, whatever I fed a compound with a freni is showing at least 3 properties. That's the information. So like this, you can see the goose rule, pepper rule, Egan rule. So all these things are implemented in our Indian database. So rule of 5 violations, 0 violation. It means that 4 out of 4 properties are getting satisfied. Okay, so that's the inference we get from impact database. And this is next database called Herb 2.0. This is traditional Chinese medicine database. Here also we can find the properties of the compounds. Other than this, we can find the drug likeness score. Here the drug likeness score is mentioned like 0.485. So that is 0 to 1. We call it as a drug likeness. Actually, we can um, term that compound as a drug. So whatever we fed with afreni showing the 0.485 is acceptable range. And the next tool what we see is the MolSort. This is not a database, this is a tool. So till now, whatever I showed, Impact and Herb 2.0 is a database, and in the database itself, we also have a tool. But this is an online tool. So you have to feed a kind of a chemical compound. Can you guess the compound? What compound I have fed at present? If you're well versed in chemistry, you might have found out. And this is a top 5 compounds, top 5 plant compounds in India. This is used for as an antiviral activity. This is also used as an anti-cancer agent, anti-neoplastic activity. Even for cold, cough, we can use anti-inflammatory. Did you identify? The name of this compound is curcumin. I think now we can able to recognize this curcumin. Curcumin is from curcuma longa plant. So according to this curcumin, you can see hydrogen bond acceptor. If you compare with the Lipinski rule of 5, they should be less than 10. Yes, it is satisfying. And hydrogen bond donor should be less than 5. Again, it is satisfying. And the log partition coefficient should be less than 5. Again, these 3 properties are getting satisfied. The fourth property is the molecular weight, which should be less than 500. So here you can see even this plant compound is satisfying all the 4 out of 4. So we can, as usual, we can tell this compound is working like a drug. And if you see this, in addition to that, we can get blood-brain barrier score 2.83 and drug likeness score. This is most important. This is what we are finding from this moles of 2. So this is a correlated formula from the physicochemical properties of the drug. If you see the drug, the drug according to this tool should be in the range of minus 1.5 to plus 1.5. We got the value as minus 0.82. So this is in this region. So our curcumin falls in this range. Just because minus 0.82 is in between minus 1.5 to plus 1.5, we can tell this compound behaves like a drug. And mole inspiration is another tool to check the molecular properties and bioactivity. The next what we discuss is the ADMET. ADMET. So before that we can also add one more thing, L. L stands for liberation. A for absorption, D for distribution, M for metabolism, E for excretion. So if you take any drug, can be a tablet, capsule, okay, if you take in the buccal cavity, the form of oral route drug delivery. So initially in the buccal cavity, so in the tongue, you can see the drug is going to liberate it, okay, and slowly it going to absorb. So the absorption is happening through the blood. So whatever drug we take, it is directly absorbed in the blood, and this will be distributed in these specific tissues. For example, if a patient is taking a drug for healing the kidney stones. If you want to treat the kidney stones, the distribution, the whatever drug we take, the drug has to be distributed only to the kidneys. So major drug has to distribute only to the kidneys, isn't it? So the distribution is happening in this process. And before and if you see the next process is metabolism. So before distribution also you can see the metabolism can also happen in between. Before reaching to the desired site, the metabolism can also happen. Metabolism is generally happening in liver. So liver is the largest, second largest organ in our human body, which comprises of maximum amount of enzymes. Okay, so we find maximum enzymes in the liver, and whatever enzymes you see, like cytochrome P450 enzymes. So these are a subset of cytochrome P450 enzymes take responsible for maximum metabolism of the drug. But if our objective is to treat the kidney stones, we should not metabolize maximum of the drug in the liver, which we call this as first pass effect. So in pharma, the much of the drug, that is more than 50% of the drug is going to metabolize in the liver just because of plenty of enzymes, these kind of enzymes present. So to overcome these kind of a metabolism, obviously we go ahead with improvising the drug delivery techniques, like using liposomes, neosomes, dendrites. So, actually, we encapsulate our drug in double lipid membrane so that lipids are not going to metabolize fast in the liver. So, some amount of drug will be coming along with the lipid, and obviously, if it reaches to the desired site, just because of change in pH, this drug is going to release, and obviously we can find the beneficial effect. Okay, so I just mentioned you as an example. And the last thing is the excretion. Excretion, the unutilized drug majorly excreted in the form of feces or urine. Other than that, even it is eliminated in the form of sweat. The excess drug is going to eliminate in the form of sweat. So what do you understand from that? Excess, it means that whatever drug is not bound to the protein, so desired target is going to excrete it. We have bound drug and unbound drug. Bound drug will give you a beneficial effect in the desired tissue, but whereas unbound drug has to be eliminated. Okay, and in addition to the excretion, we have also mentioned one more term called T. T stands for toxicity. So assessing the toxicity is very important parameter. Before 30 years, before 3 decades, we are not concerned about the toxicity. But nowadays we are focusing on each and every parameter of toxicity for the drug, whatever is marketed, whatever is marketed. So of course, if it is FDA approved, there won't be any problem. But still, we check for the long-term effects. And if you see this kind of an ADME, okay, whatever we study this LADMET, this is what we call it as pharmacokinetic study. Okay, there are actually two types of pharma. One is pharmacokinetics, which we can study that what is the effect of drug to the body, and the another is the pharmacodynamics. Okay, what mechanism of the drug, what the drug is going to act on, what mechanism? The drug is going to act on what mechanism in the body. That's what we call it as pharmacodynamics. Okay, there are different mechanisms. There are, we see all this also when we see the subsequent session. And if you see the softwares, Swiss ADME, freeware, okay, and this star drop, nine nodes, so all these things are commercial thing. So I suggest you to practice this kind of an ADME through Swiss ADME. So I just provided an input of curcumin molecule in the form of SMILES format. SMILES, canonical SMILES format, which is a 1D format of a chemical compound. This I have taken from PubChem. So whatever you see, this is a SMILES format taken from PubChem. If you feed that as an input, obviously we can get these kind of an output from Swiss ADME. So according to Swiss ADME, Okay, we can able to determine the physicochemical properties. We can determine the types of lipophilicity. We can determine the water solubility, pharmacokinetics, and also different drug likeness. So Lipinski, Goose, Weber. Here you can see the question mark, isn't it? If you click the question mark, you can see even the properties and the range of the physicochemical properties of the drug. So you have to click this. Okay, if you click this, you can even check the Goose thing. So Goose, Weber, Egan. So see, our compound is clearing with no violations. Okay, so this is something about this. But before going to this, we should also understand few more things. Where exactly you can find ADME here? I told you the ADME, studying about ADME is what? Pharmacokinetics, isn't it? So the GI stands for gastrointestinal absorption. So absorption, okay, that is most of the drug is going to absorb in the gastrointestinal tract, isn't it? If we want a good kind of a oral bioavailability, the gastrointestinal absorption should be high. So in our case, we find that is high. And the blood-brain barrier, BBB stands for blood-brain barrier. So what do you mean by this? Why we have to do this? Like if you see, the drugs are majorly classified into two types. One is lipophilic and the other is hydrophilic. So lipophilic, it means that lipid loving, and hydrophilic, water loving. So in a hydrophilic, you find much of polar groups. You can find much of polar groups. And if you see lipophilic, we have more non-polar groups. Okay, and if you see our compound, okay, is not showing blood, it's not going to penetrate the brain. The brain is more lipophilic. The brain is more lipophilic. And if you see hydrophilic, the best example we can take is the blood. So whatever drugs we take, okay, we majorly target the blood, isn't it? But if the patients are suffering from central nervous disorders or peripheral nervous disorders, then they have to design more lipophilic nature drugs. Then only it can able to penetrate the brain. So in our case, if you're simply using for treatment of antiviral infections, okay, we should get no. So our curcumin is showing no. It means that it is a hydrophilic drug. Okay, see how I am telling hydrophilic. OH groups are present plenty, okay, on the surfaces. So it means that this is a hydrophilic drug. And this contributes to the distribution. If GA absorption stands for absorption, BBB go with the distribution. And whatever you can see, the cytochrome P450 enzyme inhibitors, this stands for the metabolism. Okay, and if you see this, the final thing is excretion. The excess drug has to be excreted in the form of feces and urine, and also the it eliminates in the form of sweat. Okay, the skin permeation value is provided for this drug. Now let me go ahead with the next tool called Swiss similarity. What is Swiss similarity? In this Swiss similarity, we have to input a canonical smiles format. And if you just input as the smiles format, where you can find the Swiss similarity, this is what Swiss similarity. So Swiss ADME, Swiss similarity or nearby tools. Okay, the same URL you are going to access these kind of a tools. If you see the Swiss similarity, just inputting a chemical compound in smiles format, and I am selecting a class of compounds like a drug. So you can able to see three databases. One is drug bank, CHEMBL approved drugs, CHEMBL drug candidates in clinics. Okay, and we can see the 2D and 3D and 2D and 3D information. For example, if you select any particular thing and if you work on it, it will be getting Swiss similarity like this. So like what properties you are focusing on, okay, and what drug database you choose. Based on this, you get the similar compounds. You know, the one what query molecule I've given is the diclofenac. diclofenac sodium. I think you have heard about this kind of a drug. So when I searched this as an input, I got so many similar compounds related to this, like lumiric oxide, profan. So these are the drugs shown in Swiss similarity. So actually, I've used molecules from drug bank. That's the library I've used, and the screening method I've used is the pharmacophore. So for this, I got this kind of similarity. If you change anything else, okay, other than drug bank pharmacophore, if I choose something like a scaffold, drug bank scaffold, you may get a different kind of a results. But the most similar drugs to whatever input we have fed. Okay, so this is how we can find out of Swiss similarity. The next is the Swiss param. The next tool illustration is on Swiss param. Here, the same input, okay, just provide the canonical smiles format input here, and you can choose any of the force field. So MMFF, Merck molecular force field, Merck molecular force field base plus match. Okay, there are three methods. You can choose any of the method. So I just fitting it, and I am using a default thing, and I just click the parameterize. Then obviously what you can get, you'll be getting this kind of a result. So in Swiss parameter, we get the query whatever we fed, okay, and whatever parameters, whatever attract more closely related to the protonation state. So that will be compared, and you will be getting this kind of a result. So from this, you can able to infer the pH versus the microspecies distribution. So percentage of microspecies distribution versus pH can be determined from this. Okay, so this is about how to use the Swiss param. Now let me go ahead with the Swiss target prediction. This option is present here. In this, simply paste the smiles format, same diclofenac sodium, and then I'm going to click predict targets. So if I feed this, obviously this works similar to the target T convolution. You remember target T convolution? So if you want to work on target econvolution or reverse talking, you can also try with the Swiss target prediction. So just if you want to know the potential uh, beneficial effects of the drug. So if you see 26.7 percentage, it means that this kind of a drug is an interleukin inhibitor, interleukin 8 inhibitor. It is also cyclooxygenase 1 and 2 inhibitor. We remember on yesterday we are discussing on how to treat a common fever. In that case, we have discussed a drug called acetaminophen. So similar to that, we can use the diclofenac. Okay, so the diclofenac can also going to be working as a COX-1 and COX-2 inhibitors, other than targeting the interleukin 8 receptor. Okay, so more about the classes of receptor inhibitor information you can able to find from Swiss target prediction. The next is Swiss bioisosteer. So we can assess the bioisosterism. This is the only tool I know to work with the bioisosterism. Okay, very few have developed these kind of a tools. So here you just have to feed the structure, smiles format, okay, and you have to click the query. So the diclofenac sodium. Actually, here, if you have any isosteer, you'll be getting, or else we'll be getting this kind of a result. Some drugs, D-lactic acid, L-lactic acid, they have two types of isoforms. Okay, so if you have these kind of a more similar kind of a drugs, I mean, which is going to have more similar molecular formula, weight, etc. So that kind of a drugs you can get uh, some kind of results. But for the diclofenac, there is no such a kind of isostates available. So we are getting these kind of a result. The next tool what we discussed today is pre-ADMET. So earlier we checked with Swiss ADME for ADMET, isn't it? This is another tool, pre-ADMET. What is the speciality in this? Why we have to go through this when you already know how to use Swiss ADME? You know, here in pre-ADMET, in addition to the ADME, we can also predict toxicity. The predicting toxicity is not present in Swiss ADME. So we can able to check the toxicity in rodents like rat, and other than checking with human. Generally, the drugs whatever we use, we generally try to develop for humans, isn't it? But the veterinarians need even drugs for the specific animals, whatever they work. So we also can able to predict toxicity of rodents. Like generally, this comes under preclinical trials. Okay, so if you see that the next tool for ADMET prediction is PKCSM. Like our account, of course, the pre-ADMET. We can also use the PKCSM for analyzing the absorption, distribution, metabolism, excretion, and toxicity. So all the properties can be determined here. So this is an output. So simply feed an input. I believe I have fed an input of vitaferin. You got this kind of a result. Okay, so whatever result we got, how about the absorption, how about the distribution, this is something about metabolism, this is something about clearance, that is excretion, total clearance, renal clearance. So these values are mentioned. Different types of toxicity: AMS toxicity, HERG toxicity, oral rat toxicity, hepatotoxicity. So ADMET properties can also be predicted from PKCSM, a single tool. So this is another kind of a website URL to work with it. Swiss target prediction, I think I already explained you. The next thing will be seeing is the Protox 3.0. This is used for prediction of various types of toxicity. So 2 years before we had 2.0, now it is upgraded to 3.0. And if you see here, we have to feed an input of PubChem name, that is NCBI PubChem ID, or you can simply give an input of a keyword of a drugs or plant compounds, whatever you know. Okay, so or you can also feed an input of a smiles format, and if you simply search, okay, so this is the tool actually for predicting the toxicity prediction in Protox 3.0. So if you click this, okay, and if you feed the smiles format, we can get these kind of properties to be selected. For example, in my research, I am focusing on hepatocellular carcinoma, and I'm using the primary target as heat shock proteins. So in that case, first we have to check whether is it hepatotoxic or not. Definitely, whatever drugs we check for hepatotoxicity, we must check for nephrotoxicity because excess drugs has to be eliminated from kidney. So if kidney is going to take much of the drug, it can also harm the kidneys. So yeah, whatever drugs you design for hepatic properties, we should assess even the nephrotoxicity and cardiotoxicity. These are the default things we have to select. But nowadays, few drugs also gives respiratory toxic, so we can select these also if we need. And then my major target receptor is, of course, a kind of mitochondrial protein. So I can select this. And if the carcinoma HCC, the major target protein is P53, no doubt. So I am selecting this also. Like this, we can select whatever we want. And then we have to simply click Start Toxicity Prediction. Whatever we have selected, we'll be getting the results based on that. Okay, so mall inspiration window. So this is a ACD chem sketch. This is a chemo informatics tool. So if you want to download 3D structures, or if you want to find the compounds, draw the compounds and get the smiles format, everything is feasible with this ACD chem sketch tool. This is a freeware. Download and install, and you can use this as a visualizer for the chemical compounds. ACD Chem Sketch. No, this is not ACD, but this is Vega tool. This is not ACD. This is Vega. So in Vega, we can also design the peptide. If you want to design a peptide, okay, this is a freeware. Install the Vega tool, and if you open this, okay, you'll be getting this kind of a window for building a peptide. Simply you have to click these buttons to get this amino acid sequence. And then if you click build, you get a nice peptide in 3D structure. You can save this PDB structure. We also have an online tool called PepFold 3. This is used for building the peptides. Okay, so simply you have to feed the amino acid sequence, and then if you click run, obviously we'll be getting this kind of a 3D structure of peptide. To download this, we can go to model1.pdb. This is the option. If you click this, we can download the PDB of peptide. The next we'll be discussing is drug-drug interactions. See, till now we have discussed on how to assess the drug likeness and how to assess the ADMET properties. Okay, so other than this, we also discussed on how to model the chemical compounds if the structures are not available in PubChem, and how to model the peptides if these peptides are not available in databases. The next thing we'll be discussing is what is the effect of drug on the other drug. Generally, if you see asthma patients, if they feel arthritis, whether whatever drug they take for arthritis can contradict this kind of can give any kind of adverse effects with the existing asthma drug or not. So many of the asthma patients try to take homeopathy medicines. So in that case, it gives some complications. To check the multiple drug combination effect reactions or even adverse reactions, we can check from a tool called drug-drug interactions from drug bank, DDI checker. Here in this example, I have checked with acetaminophen with warfarin. Acetaminophen, this is to relieve the pain, paracetamol. We take to relieve from pyrogens, that is the foreign matter which is pyrogens. Okay, that is to relieve from this foreign matter which is causing the high temperature, and also to relieve from pain we are using acetaminophen. Warfarin is a blood clotting agent. Generally, the warfarin, the name itself implies war. So during the war, in the Second World War, many got injured. So they are given a drug called warfarin for the which act like an anticoagulating agent. Okay, the acetaminophen and warfarin. This has a moderate kind of an adverse effects, but some drugs in combination give severe adverse effects. Okay, so that should be avoided. So drug-drug interactions can also be checked through drug interaction checker from drugs.com. Okay, and this is the input window. And the next thing, food drug interactions can also be checked from drug bank through a tool called food interaction checker. See, that's also most important, isn't it? Just designing a drug, we can't tell that it's going to cure a patient. Even if it can cure a particular disease, it may lead to another disease. For example, whatever I worked in my research project, okay, whatever I got the grant, okay, that also I later I came to know that it's going to give some kind of an adverse effects. So first we have to check all these kind of a parameters, okay, in designing the drug. I give you that kind of an examples when I start taking few more sessions. Okay, food interaction checker. Here, what exactly I checked. I checked the effect of the different vitamins. Okay, if I take a vitamin tablet, multivitamin tablet, whether I can able to take acetaminophen, and also whether I can able to take warfarin or not. If you see, okay, only one thing, the cyanocobalamin, like the cyanocobalamin is majorly present in liver. If someone is going to take the liver food, they should not, if they are taking acetaminophen and warfarin, they should not take liver. They should not consume liver, mutton liver or chicken liver, or even they should not take this kind of cyanocobalamin enriched vitamin tablets. Okay, so this is something we can check even food drug interactions. And the vitamin A is not giving any interaction paradoxon. but try to avoid alcohol. If consuming alcohol can increase the risk of hepatotoxicity. So these kind of information also we got from that. Foot drug interaction checker from drug bank. The final tool on today's session is reverse talk. In reverse talk, okay, this is the URL reverse talk. Simply input a ligand, and then you can find a series of receptors, like however you find, and you can also find the binding energy score. Okay, so for this we have to try with this URL mentioned below. Okay, now let me go ahead with the today's practical sessions. Say, I think we have come across numerous information, isn't it? Can we check one by one? First, we go with the PubChem to take the chemical compound. Any doubts you can ask me before taking you to the hands-on sessions. Cholesterol. What is that cholesterol? Want to ask any questions? You can ask me by unmute and ask, or else I can take you directly. So, can we take and download one plant compound, quercetin? I told you, no, for I worked with a compound called quercetin, which is curing the ovarian cancer. Can we try the same? Quercetin. Okay. So just type the quercetin in PubChem, NCBI PubChem. We got this. We have to get inside. So to feed an input, okay. So we can take even the smiles format. So to take the smiles format, I'm just dragging down, checking for the smiles format. See, I got the smiles format now. So just copy the smiles format. In reverse stock, we have, so home, reverse stock home. Click submit. So here it's asking for mol2 format. So the smiles format, whatever we take, this can't be fed as an input, but it's asking for mol2. Then what to do? What to do? We have to download the 3D structure. To download the 3D structure, go to the 3D conformer, download coordinates, STF. So whatever I'm going to save, that quercetin, okay, whatever I saved is initially in the form of .stf format, but I have to convert as .mol2. To convert as mol2, you can use OpenBabel or Babelwin softwares for file conversions, or else simply open your compound, that is quercetin compound, in Discovery Studio Visualizer. Click Save As. In Save As, we have Cybel mol2. Select that and then give quercetin.mol2. Now our compound quercetin, this is our quercetin saved as mol2 format. So just go here, you can see quercetin.mol2 is available. So we have to go to the reverse stock, browse that, and you have to give like quercetin.mol2 and then click next. It's asking for PDB files or alpha fold. So if you want to check the reverse stocking with multiple receptors, go with either PDB file, give the PDB IDs or give the alpha fold uniprot IDs. If I click alpha fold, I was asked to enter the uniprot IDs. If you type PDB, it asks you to give only the PDB as an input. Here, this tool is developed to only for these species: human, E. coli, mouse, fruit fly, etc. Okay, very few species only there we can able to find in this tool. So I better try with the human, mice, and rat. So for this, I'm going to the uniprot. So I try to take the trap one. The trap one uniprot ID is this. Okay, I'm copying and pasting as a uniprot ID. Press enter. And now I go with human uniprot ID. 6-letter ID is this. Copy and paste and enter. And the next is the mouse trap one. You can see trap one mouse, trap one human, trap one rat. Okay. So now I'm going to paste this trap one mouse. So that's it. You have to simply check the yes, out of 3 provided receptor, 3 can be docked. We got this because this is taken from the 3 different sources. Click next. It's asking for email ID. Provide the email ID. Submit. So immediately you won't get the result. It takes some time. Okay. So after the results are generated in your email ID, you can open and see. Meanwhile, I can take you to the next tool called Swiss ADME. So in Swiss ADME, we have to feed the canonical smiles format of the chemical compound. So I go to the Swiss ADME and then I type this smiles format, not this smiles format. The smiles format is available here. So is available here. Copy this smiles format and paste it here. Now you click run. So I increase the size of the window. So now whatever we are trying is from Swiss ADM. In Google, if I type Swiss ADM, it directly takes you to this URL. So after Swiss ADM, you will be trying with other tools also. We got the result. We are getting the result loaded. First, we can see this result. Show boiled egg diagram. The one what I searched is a quercetin. The quercetin is present in, this is a boiled egg diagram. The white part shows the albumin and the yellow part shows the yolk. The albumin is hydrophilic in nature, but the yolk is lipophilic in nature. So if our drug, this red color, is present in this location, yellow color, we can tell it is a lipophilic drug. But this is present in white color region. That too, it is having so much distance. So it is more hydrophilic. See, if I click this, you can see so many kind of OH groups. So our compound has so many OH groups. So only we got like a more lipophilic, more hydrophilic in nature. Okay, so this is what you can infer from this. If you feed an input of acetaminophen, if you feed the input of acetaminophen, that is your homework today. You can try that. Tomorrow I'll be asking you whether it is showing a lipophilic or hydrophilic. Okay. So next, I just going to discuss on quercetin. This is the quercetin. So here you can able to see the molecular weight less than 500, the hydrogen bond acceptor less than 10, donor less than 5, polar. Next is the partition coefficient less than 5. So all the four properties are satisfied. So Lipinski zero violation. Okay. So Lipinski can see not only less than 5, less than or equals 500, less than or equals 10. So even this is less than or equals 5, so only you are getting zero violation. Similarly, if you see the goose has different properties. The molecular weight should be between 160 to 480. 160 to 480. And partition coefficient is in between minus zero point -4 to 5.6. And molecular refractivity, this is an additional term. This is an additional physicochemical property of the drug. The molecular refractivity should be in between 40 to 130. So like this, we can see different properties in different drug likeness filter. And ADME, that's what the major thing we are discussing. The G absorption is high. So better it can be used as a oral drug. So BBB, no permeate. It means that it's not going to give any adverse effects with respect to central nervous disorders or peripheral nervous disorders because it's a hydrophilic drug. And we can see the metabolism and excretion value. Skin permeation value is minus 7.05 centimeter per second. So this is how we can assess the results of ADME, that is pharmacokinetic property. So other than this, the bioavailability score is mentioned is 0.55. Okay, the next what we are going to try is Swiss parameter or Swiss similarity. There is a Swiss similarity. Yes, we go with the Swiss similarity. So in the Swiss similarity, if the server is not working, you can try on some other day when the server works. Presently, the server is not working. Okay, so you can try on that. Swiss parameters. So we have to simply feed the canonical smiles format. The default thing is mentioned here. Okay, simply we have to select and draw a molecule using the sketcher. So I simply click enter. We are getting the kind of a results in the Swiss parameter. Okay. Meanwhile, I can take you to the next. So this is the Swiss parameter. For the quercetin, whatever I searched, okay, we got a related kind of a compounds. A slight variation you can see. O, OH. Here you can see OH, O minus. So related compounds and that related compounds kind of an microspecies distribution versus pH. You can able to see here in Swiss parameter. Okay, the next what we discuss is the pre-admet. Pre-admet. So this is another tool for assessing the ADME. Other than ADME, you can also assess the toxicity. So to assess the toxicity can be Ames test or rodent toxicity. We can go with toxicity prediction. So I'm just getting inside this. Either it asks for Smiles format or you have to upload the Mol2 format. Okay, so I'm going to click this. It's asking like .mol. See, it's asking to paste the .mol, not .mol2. So how to get .mol file, save as. So it can, yes, we have MDL mol. So select this .mol. So now I save the same compound as .mol. So whatever I saved, okay, just open the folder. So where is my quercetin.mol? This is my quercetin.mol. Open in Notepad or Notepad, whatever editor we have. No, no, it's opening in ASD Chem Sketch and defaultly. So this is ASD Chem Sketch actually. If you want to, I'll be explaining more about it. Yeah, yeah, excuse me. Kindly unmute. I mean, kindly mute. Okay. See, this kind of disturbance should not be there. So take care when you log into the session. Okay. So I think if you have any doubts, you can ask me. So now I'm going to open the folder. Persian.mol is directly open in SDKM sketch, but I want only the coordinates. The coordinates I can open in Notepad. So whatever coordinates I have .mol, just copy the entire coordinates .mol. So whatever I copied, I pasted here. Okay, pasted here and click load. So we got this chemical compound loaded. Or else you can draw this using the tools available. Click submit. So whatever I'm going to upload it, that is quercetin, I'm assessing the toxicity using the pre-admit. The next tool what I'm going to show is ProTox 3. To assess the toxicity, we can use this. So we have to go to the toxicity prediction. The first window I click, type the canonical smiles. Take the canonical smiles, paste it, smiles, click, then only it is going to change here. So this is our quercetin compound. So after the track down, I'm going to check with hepatotoxic, nephro, cardio, and carcinogenicity, immunotoxicity, mutagenicity, cytotoxicity, and then mitochondrial membrane potential, P53, which is of course a most important target receptor for ovarian cancer. So these are what I have selected. And if you want to select any of these molecular initiating agents, you can select it. Then click start prediction, toxicity prediction. This is most widely used tool nowadays to determine LD50, lethal dose 50. Like if you want to work on Small animals like mice and rat, it's a must to work with this tool to check. Okay, this has saved us time even when my PhD scholar started working on the effect of anti-cancer drug on mice. So whatever we got, we just taken this kind of a drug dosage and we just treat it. We don't find any toxic effects. Okay, if you see this compound results, okay, you can see it bottom. See, in our case, whatever effect quercetin is showing inactive, it means it can able to cure hepatic related problems, but nephrotoxicity. Quercetin, if you take too much tomato, what is going to happen? You can find kidney stones formation, isn't it? You accept this? I think many of them know this also. But tomatoes can cure ovarian cancer, but excess consumption of tomatoes can lead to nephrotoxicity. But cardiotoxicity, no, nothing, nil effects. Carcinogenicity is there. We should not exceed the preferred dose. If we exceed, then it leads to the carcinogenicity. Immunotoxicity, inactive. So like this, you can see, okay, about so mitochondrial. It's going to target the mitochondria, the quercetin. So like this, you can find this information, and this is the pre-admit toxicity. Okay, so I'll get toxicity, carcinogenicity in rat, carcinogenicity. See that quercetin is carcinogen for rat, but that is not for mice. So it depends. In protoxity, whatever we predicted is for human. Also, that is used for prediction of rat. So whatever we have found milligram per kg is for rat. Okay. So like this, we can able to assess the kind of pre-admit properties using this, and drug likeness properties. I told you how to assess, and we are one more tool is there, mole soft and mole inspiration. Using this, we can also assess the drug likeness values. Okay, mole soft and mole inspiration. So mole inspiration is of course a chemoinformatics tool. You have to go to the tool, and where is my option? So here also you can evaluate calculation of molecular properties. So feed the Smiles format, and then we can able to calculate, or you simply draw the compound. Okay, so I just drawn the phenol compound, calculating the properties. So we'll be getting the properties for that. So this is how we can use the online tools. Okay, so I have completed the illustration of the one more thing I left out is the drug bank. Drug bank. Can we go with the drug checker, drug interaction checker? So the drug interaction checker of drug bank. So first I'm going to type one kind of a drug like quercetin. Quercetin is available. Then the next thing what I'm going to try is acetaminophen or warfarin. Warfarin, sodium. Check interactions. So major, the metabolism of warfarin can be decreased when combined with quercetin. So it gives a kind of an antagonism. Okay, so it should not we should not use. This is not going to give a synergistic effect. That's what you understand. Okay, you check with some other drugs. Okay, the warning also you have to see. Similarly, you can try with food drug interactions. Okay, so simply there we have to try drug bank, food drug interaction. So food interaction checker. Here you can give the food name like vitamin A vitamin. We can try with warfarin, like whatever foods rich in vitamin A and warfarin. So no food interactions. So like this, you can. Okay. So I've completed today's session. Any doubts I can ask. If anything left out, I'll be teaching you on tomorrow. Any doubts? If no doubts, thank you very much for the patience. And also, I'll be circulating a feedback form. Kindly provide your valuable feedback. If any corrections has to be, if anything I can able to implement in my pedagogy, I can definitely implement it.
Sir, one last thing. Today it was really good session, and the websites that you have shown it to us. Can we get the list of something like that in tomorrow's session, or by email, or by any other means?
So actually, your voice is not
Oh,
clear. So whatever you
okay.
want to mention, you can message me in the WhatsApp in the form of a
Sure,
WhatsApp message. And you know,
sure.
actually, in the WhatsApp group, I have already pasted the URL of the tools and databases on yesterday. If you go to the tools and databases, if you are asking that, you can able to find in the yesterday's message.
No,
You can,
I was asking about the today's session. That whatever the.
yeah, yeah. This video I'll be sending you. I'll be
Thank
sharing
you,
the
sir.
video. Yeah, yeah, sure, sure.
Thank you, sir. Thank you so much.
Yeah. So, any other doubts? So, okay, very good. Thank you very much for the interaction and patience maintained in the session. Have a great day to you all.